Hello students, today we'll see the next topic for the different methods for the estimation of the proteins. In the previous lecture we have seen the Lorry method, then different dye binding methods such as the anionic dye binding, then Bradford dye binding methods for the estimation of the proteins. Today we'll see the Bicinchoninic acid method, and the second one is the UV absorption method at 280 nanometers. So let us see one by one. So the first one is the Bicinchoninic acid method. So here what happens, the proteins and peptides, which are if you are considering the two bonds, we can say the dipeptides are, whenever they are in contact with cupric ions, they will reduce cupric ions into the cuprous ion under alkaline conditions, and which is similar in principle that what we have seen in the Biot reaction. So the cuprous ion then reacts with apple greenish bicinchoninic acid reagent to form a purplish complex. So one cuprous ion is chelated by the two BCA molecules. So that particular reaction, which is carried out under alkaline conditions and forms the cuprous ions, and it gives the purple color, and we have to estimate that purple color intensity at 562 nanometer. The color measured at 560 nanometer, which is a near linearly proportional to the protein concentration over a wide range of concentration from micrograms to 2 mg per ml. So here we have to determine the concentration at wide range of the concentration, that is a microgram to mg, that is up to 2 mg. So the peptide bonds and 4 amino acids such as the cysteine, tryptophan and tyrosine, these are contribute for the color formation whenever they are reacted with the bicinchoninic acid and forms a purplish complex, that is the one cuprous ion is chelated by the two bicinchoninic acid molecules. Now what is the procedure? So first we have to in the first step we have to mix the protein solution with a BSA reagent which contains the BSA sodium salt, sodium carbonate, NaOH and sodium sulfate under alkaline condition. It means that the pH of the solution is around 11.25. Then we have to incubate it at 37 degrees Celsius for 30 minutes or room temperature for 2 hours or 60 degrees Celsius for 30 minutes. The selection of the temperature depends upon the sensitivity desired. So the higher temperature gives the greater color response, and accordingly we have to use the methods. Now read the solution, read the absorbance of the solution at 562 nanometer against the reagent blank. We have to use the reagent blank for the particular estimation. Then construct a standard curve using BSA. As usual, we have seen in the previous methods, we have to use the bovine serum albumin standard, and we have to construct a standard curve, and from that we have to find out the amount of the proteins present in the sample. Now what are the applications? So the BCA method, that is a bicinchoninic acid method, is used in protein isolation and purification. So what are the advantages? So this method having sensitivity which is very comparable to the Lorry method. It is sensitive to the micro level BCA, that is up to 0.5 to 10 microgram, which is better than that of the Lorry method. So one step is a mixing which is easier than the Lorry method. So the reagent is more stable than for the Lorry method. As we have already discussed about the Lorry method, we have to use freshly prepared phenol reagent for the estimation because it is not stable reagent. So non-ionic detergent and buffer salts do not interfere with this reaction. Medium concentration of the denaturing reagent, that is 4 molar guanidine hydrochloride or 3 molar urea, do not interfere in the analysis. So these are the advantages. Now what are the disadvantages? So the color which is obtained, it is not stable with the time. So we have to take reading very fastly. So the analyst needs carefully control the time for the reading absorbance. So any compound capable of reducing copper 2 plus to copper 1 plus will lead to the color formation. So this is one of the disadvantage of this method. So the reducing sugar interfere to the greater extent than in the Lorry method. So the high concentration of the ammonium sulfate is also interferes. So as we know that we have already discussed in the the chapter carbohydrate, we have know that reducing sugar will reduces copper. If the food sample containing some amount of sugars which are reducing in nature, which will cause the formation of conversion of cupric to cuprous is going to take place, and at that time there is a formation of some kind of color and it will interfere in the results. So this is one of the disadvantage. So color variation among proteins are similar to those in the Lorry method. Here also we have to get a color variation due to different nature of the proteins. Now the next method is UV, that is ultraviolet 280 nanometer absorption method. So look at what happened here. We have to measure the absorbance with respect to that particular wavelength, and by which we have to find out the concentration. So generally what happened, protein shows a strong absorption in the region at ultraviolet at 280 nanometers. So the primarily due to presence of tryptophan and tyrosine residues in the protein, it will shows the absorbance at that particular range. Because of the content of the tryptophan and tyrosine in the protein from each food sources is fairly constant, the absorbance at 280 nanometer would be used to estimate the concentration of the proteins using as usual Lambert-Beer's laws. Since each protein has a unique aromatic amino acid composition, the extinction coefficient, that is the epsilon, at 280, or we can say the molar absorptivity at 280 nanometer, must be determined for individual proteins for the protein content estimation. Then what is the procedure? So first of all, what we have to do, we have to take a proteins and which is solubilized in a buffer or alkali. Then we have to take absorbance of protein solution and add 280 nanometer against reagent blank. Then protein concentration is calculated according to the usual equation, that is Lambert-Beer's law. A is equals to ABC, where A is the absorbance, small a it is the absorptivity coefficient, B is the path length or we can say the cell arc weight, and C is the concentration. So now what are the applications? So the UV 280 nanometer method has been used to determine the protein content of milk and meat products. It has not been widely used in the food system. So this technique is better applied in the purified proteins system or the proteins that has been extracted in alkali or denatured agents such as a 8 molar urea. So what are the advantages? So it is the rapid and relatively sensitive method. At 280 nanometer, 100 microgram or more protein is required, several times more sensitive than the Biorate method. Here, no interferences from ammonium salt and other buffer salts also. So this method, it is a non-destructive. Sample can be used for the other analysis after protein determination, and this method is used very widely in the post-column detection of the protein. Then what are the disadvantages? So the nucleic acid also absorbs at 280 nanometer. So the absorption 280 nanometer divided by the 260 nanometer ratio for the pure protein and the nucleic acids are 175 and 0.5 respectively. So what happened? The ratio for the pure protein and nucleic acid, it is of 175, and for the nucleic acid it is of 0.5. So one can correct the absorption of the nucleic acid at 280 nanometer if the ratio of absorption of 280 nanometer and 260 nanometer is known. So accordingly we have to correct that. So this is one of the disadvantage, that nucleic acid and that particular amino acid, that is a pure protein, also showing the same absorbance. So the aromatic amino acid contains in the proteins from various sources differ considerably. So what happens as soon as there is a ring structure is increases, so accordingly chromophoric groups are altered, or we can say due to presence of a long ring structure there is a difference in the lambda max. So we can say that it shows the different kinds of colors. The solution must be clear and colorless, so the turbidity due to particulate matter in the solution will increase the absorbance. Okay, so the solution is clear and colorless. If the solution is colored, then there is a no such interference of turbidity which is occurred due to some kind of particulate matter. This method, a relatively pure system is required to use this method. So pure system is required for using this method. So this is all about the methods used for the analysis of proteins. So in the next lecture we will see the protein characterization such as the protein efficiency ratio and other kinds of things we will see in the next lecture. Okay, thank you.